Hello, everyone. My name is Kristen Milbury, and on behalf of the Foundation for Conservation of Atlantic Salmon, I would like to welcome you to today's webinar. If you wish to use translation, please navigate to the chat where you'll find instructions on how to connect to AI translation that we have available. For today's webinar, we are very pleased to be hosting Dr. Bobby Nakamoto. Originally from Northern California, Bobby acquired a Bachelor of Science in Environmental Systems at the University of California, Merced in 2015. He extended his undergraduate research into a PhD in ecology, evolution, and organismal biology, during which he studied the trophic ecology of Chinook salmon using bulk and compound-specific isotopic methods. After completing his PhD, Bobby served as science manager for the University of New Brunswick's Stable Isotopes in Nature laboratory, where he supported lab operations and developed isotopic methods for tracing seafood catch provenance, among other things. In September of 2024, Bobby started his current job back at UC Merced as Director of the Office of Research and Economic Development's Environmental Analytical Laboratory. Today, Bobby will be speaking on Stable Isotopes in U, a primer for environmental and ecological research. After this afternoon's presentation, we will open the floor for questions and answers. I will now turn the webinar over to Bobby.
Great. Yeah, thank you very much for the introduction, Kristen, as well as for the invitation to be here. I'm really pleased to have the opportunity to talk about stable isotopes, a tool in ecology, environmental studies, and really any type of science or any type of investigation that I think is really powerful and really cool and fun, because it gives you a chance to be really creative with the kind of problems that you might be facing in whatever kind of research you're trying to do. One anecdote that I like about stable isotopes is from a professor who's taught a stable isotope course for a long time. And he was of the impression that at the very beginning, you mention stable isotopes to people and they sort of, their eyes glaze over and it's, what could that possibly be useful for? Why do I care? What is that? And hopefully by the end of the course, many people feel like it's very easy to think of things or ways that isotopic information could contribute to what you're wondering about. And it's very difficult to imagine a problem that stable isotopes could contribute nothing to. So without much further ado, I'll get started here. This may be, or this next slide here. This may be a slightly more broad talk than some of the other webinars that you've been to in this series. I looked at a couple of them and many of them were very specifically related to Atlantic salmon. It's my target here to just give you a sort of broad introduction to stable isotopes and really tie that into maybe some of the kinds of problems and systems people that are interested in Atlantic salmon or aquatic ecology might care about. But in general, it'll be very broad and just sort of isotopically involved. So again, circling back, it'll be an overview here. What is a stable isotope? That's where we're going to start. Stable isotopes are this weird abstract thing. We talk about them using some strange terminology, some funny Greek symbols. It's my hope that by the end of this presentation, you'll know what you're looking at a little bit when you see some of that stuff. I'll follow that up with getting into just some of the mechanics behind common isotopic systems. So in this presentation, I'm going to focus on carbon and nitrogen, because they're probably the most common isotopes you're going to encounter out in the world, out in the scientific literature. So it's a really good place to start. And if you have a good foundation in those, it becomes relatively easy to start understanding where other isotopic systems might be able to contribute to the questions you're trying to ask. And lastly, I'm going to finish with something I've sort of cheekily called isotopes, where I'll discuss a little bit about the differences in isotopic compositions you might observe if you're looking at different tissues, or if you're looking at different tissues even within a single individual. So why might we analyze different parts of an organism, and what different parts of an organism might we analyze in the first place? So with that, I will jump into what is an isotope. And again, this will hopefully be a necessary minimum. In general, isotopes are atoms of an element with a differing number of neutrons. So some of you may remember from introductory chemistry classes that when we think about atoms of an element, there's really three main constituents. We have electrons, which are negatively charged, floating around out in these orbitals around a nucleus. And the nucleus is the core. Most of the mass of an atom is in the nucleus, and the nucleus is primarily made up of protons and neutrons. The number of protons that you find in the nucleus generally relates directly to the atomic number in the periodic table. So the number of protons is determining what element you actually have on your hands. 1 proton, it's hydrogen. 6 protons, it's carbon, and so on and so forth. The neutrons are a neutral particle, so protons are positively charged, electrons negatively charged. The neutrons are neutrally charged, and they help hold the nucleus together and keep the atom functioning as a whole. But you can have variable numbers of those neutrons. So most atoms, the most common forms of elements, have the same number of neutrons or a similar number of neutrons in the nucleus as protons. The heavier elements, that differs a little bit, but things like carbon, nitrogen, oxygen, the stuff that's really common in organic matter, you generally have the same number of neutrons in the nucleus as you have protons. But occasionally you have an atom that comes along that has an extra neutron in there, and that increases the mass of that atom just very slightly. So I'll use an example here for carbon. For carbon, the most carbon in the world is carbon-12, 6 protons and 6 neutrons, 98.89% approximately. There's approximately 1.1% of carbon that has an extra neutron, so that would be carbon-13. And carbon-12 and carbon-13 are what we call the stable isotopes of carbon, meaning that they were formed, early on, probably in the core of a star somewhere, and they're not going to be changing at any time, unless you put them in a particle accelerator, in a nuclear explosion, something of that nature. The amount of carbon-12 and carbon-13 in a sample will be the same no matter what you do. It is a stable isotope and won't be changing. Carbon also has a radioisotope, we would call it, or an unstable isotope that will radioactively decay. This is carbon-14, so it's a carbon that has 2 extra neutrons. And carbon-14 will radioactively decay into a stable isotope of nitrogen with a half-life of about 5,730 years. This presentation will not be concerned at all with carbon-14. It's so it's not abundant enough to be really important for a lot of ecological environmental questions where we're using stable isotopes. So stable isotopes, we focus only on the stable isotopes, as you might imagine. So of course, part of the whole point of stable isotopes, and it should come as no surprise to you that at this point I've said that there's 2 types of carbon, the stable and the lighter and the heavier isotope, carbon-12 and carbon-13. Everything that has carbon in it contains a mixture of these isotopes. This is a diagram here on the right that I really enjoy, and I can't remember exactly what the total mass of this person is, but this is the hypothetical isotope person. So in any given individual at what we call natural abundance, so we haven't been adding any sort of stable isotopes, this is just what you would find in a normal person who's been going around eating normal things, they'll have about 11.4 kilograms of carbon-12 in them and about 137 grams total of carbon-13. And you can do this for other stable isotopes. So we see his hands are sulfur-34 and sulfur-32, with generally there being more of the lighter stable isotope, about 220 grams of sulfur-32 and about 10 grams of sulfur-34. And again, part of the utility that we take for granted in stable isotopes is differences in the abundance of those 2 isotopes. They mean something to us. We can analyze them and learn something about the thing that we analyzed. But how do we actually discuss those differences? Do we go around talking about, oh, they had 10 grams of sulfur-34 in them? Not so commonly. Most stable isotope scientists are going to be relating their data to one another's using what we call delta notation. So hopefully, again, not everybody's eyes are glazing over now that I've thrown an equation on the slide here. Don't linger on it too much. The most important thing for you to take away about what delta notation is telling us is, number one, an important thing to know about delta notation, I should circle back this, is we're always comparing stable isotopes to an international reference material. This is something that all stable isotope labs and all stable isotope practitioners around the world have agreed upon. There's a set number of reference materials that we're always referring to. So for carbon isotopes, we are always reporting our data based on whether or not the sample that we analyzed in lab has more abundant or less abundant heavy isotope than what we call Vienna PD belemnite. The original PD belemnite was a squid-like animal. It was a fossil that we analyzed, and we used it to establish the scale. We ran out of the fossil because we analyzed it all. So the International Atomic Energy Agency in Vienna, they basically created a new ratio that we use as the international reference material. So when we analyze a sample, we get the ratio of the carbon-13 to the carbon-12 in there. We put it into the delta equation, and we get out a value like, say, minus 27. And that's in terms of per mil. We say delta 13C would be minus 27 per mil. And per mil is a word very much like percent, but the mil stands for 1,000. So we can relate this somewhat in percent terms. A negative per mil value means that there's less of the heavy isotope in the sample than there was in the standard. A negative value of, say, minus 27 means that there was about 2.7% less of the heavy isotope in the sample you analyzed than there was in the standard. If that value had been positive, there would have been 2.7% more of the heavy isotope in your sample than there was in the standard. And that was all relatively complex. I explained it in sort of a winding fashion. Hopefully you sort of are understanding what I was driving at the end. But at that point, your question is probably why bother with that complex delta notation? Why aren't we just reporting ratios directly to each other? We can measure them. Why don't we just use, the ratio of the carbon-13 to the carbon-12? And the reason for that is somewhat twofold. One really good reason is when we actually calculate those ratios or actually measure those ratios, you get numbers like you see here on the bottom of my screen, 0.011202 versus 0.01118. And that just doesn't really have a very nice ring to it if we're trying to compare those numbers in a presentation like this. It's much easier for me to say something like minus 27 than go out to 6 or 7 decimal places in a fraction like that. So using delta notation makes all the numbers that we talk about and discuss for our data much easier. The other thing is while we can do it effectively, it is difficult to measure stable isotope ratios. And so by always relating the amount of isotope in our sample to a reference material, it makes our measurements more accurate. You can think of it as if you were trying to estimate the heights of a bunch of people on an NBA team, you might look at them and say, well, they're tall, I don't know, 6 something, 7 something, I'm not sure. But if you were given, a reference person and all you had to do was say whether or not everybody on the team was taller or shorter than your reference person, you would be pretty accurate at being able to do that. So by always including a reference material, we're able to make what is in effect a pretty difficult measurement very precisely and very reliably, and it makes all our data much more comparable in the long run. Okay, so I think I've covered, again, a necessary minimum of what you need to understand to at least look at a bit of stable isotope data. I'm now going to move forward into, what are we doing with stable isotopes? What's the utility? What are they doing out in the world? There's carbon-12 and there's carbon-13. I've told you they're basically exactly the same except for that extra neutron. What matter does it make? And of course, ecology, environmental sciences have been described as the economics of nature. Most of the big questions that ecologists and environmental scientists are asking themselves ultimately boil down to things like what stuff is going where, how much of it is going there, when is it going? And it's lucky for us that when we're trying to do accounting like this, we have some conservative tracers in the form of stable isotopes. About 98% of ecologically relevant materials, so that, that's organic matter that's, in animals that we care about, in plants that we care about, that stuff is made up of about 98%, like I said, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, the schnapps elements, some people might say. For all of those elements except phosphorus, which unfortunately, I dream about a world where phosphorus has another stable isotope that we could use to learn cool things about what phosphorus is doing in the world. All those other elements have more than one stable isotope. So all those elements have a tracer that we can use floating around in the form of that heavy isotope that is a small fraction of the carbon, hydrogen, nitrogen, oxygen, sulfur that we care about in the world. So how does this tracer do useful things for us? We know that there's 2 types of most of the elements we care about. We can measure them, but how are they being partitioned and how are they actually giving us useful information? So the next piece of this puzzle is understanding, again, I've told you that the 2 isotopes of carbon, I'm going to continue to use carbon as an example here. They undergo the exact same reactions. They're both carbon at the end of the day. So a plant that is trying to take up CO2 to do photosynthesis, it doesn't really differentiate between the carbon-12 and the carbon-13. It just sees CO2 that it's taking into its cells and it wants to do photosynthesis on. So you can imagine these 2 isotopes, the carbon-12 and the carbon-13, as being Lego blocks that are the same exact shape, but a different color. So if you were, say a child or, even a grown person building a Lego structure, you could build the same exact structure, morphologically using the Legos. But if you used a pile of blue Legos and a pile of red Legos to build 2 identical structures, those 2 structures will still look very different to you if you know how to look. And so to extend this metaphor a bit into kind of a natural system, we can think about Rubisco. Here we have a 3D model of the enzyme Rubisco. For those of you who aren't familiar with it, Rubisco is the enzyme that's responsible for carbon fixation in photosynthesis. So Rubisco will be taking CO2 and sticking it into fixed carbon compounds, making it organic matter that organisms can make use of. And I said that the plants really don't differentiate between carbon-12 and carbon-13, but that's not exactly true. For reasons that are a little bit more complicated than I want to get into right now, chemical processes can be thermodynamically choosy. So in effect, you can have an enzyme, a process, in this case, an enzyme like Rubisco, which maybe prefers blue Legos. We'll say it has a preference. It finds it easier to work with the blue Legos. And there's some reason for this on the right. You know, light isotopes move a bit faster, they diffuse a bit faster, they form slightly weaker bonds. So for, again, reasons like that, reactions can have a preference. So if we started with atmospheric CO2, a big pile of red and blue Legos floating out in the world, and we let our friend Rubisco loose on it, what we'll see is on the left here, Rubisco is responsible for carbon fixation or photosynthesis in a group of plants that we call the C3 plants. We call them that because what Rubisco initially does is it adds the CO2 it takes in to a compound that has 3 carbons in it, hence the C3. And we know that the big pile of red and blue Legos that's floating out around in the atmosphere, it has a delta 13C value of about -7. So the original playpen full of Legos is the atmospheric CO2. It has a value down around -7 or 8. And choosy Rubisco selects mostly blue Legos. So in this situation, the blue Legos are the lighter isotope, carbon-12. And so it takes on, when it fixes carbon into its tissues, a very negative value, about -27 per mil relative to the original carbon it was working with. So an original pool of red and blue Legos, Rubisco has chosen to select mostly the lighter isotope, such that now the organic matter that we see in the plant that the Rubisco was acting in now has less of the heavy isotope percentage-wise than the original pool does. If we contrast what Rubisco does with something, a different enzyme, so a different person building a Lego structure, we can consider the enzyme PEPcase, that's PEP carboxylase. It's the acting enzyme in a group of plants that we call the C4 plants. It's a water saving strategy for some plants to use this kind of photosynthesis, helps them thrive in warmer environments than C3 plants. You don't need to care about that right now. All you need to know is, in a general sense, you could imagine it as PEPcase is slightly less choosy. It doesn't mind if it takes up a couple red Legos for a couple reasons. So if we compare the work of C3 plants and the work of C4 plants, we find that C3 plants have different carbon stable isotope compositions than C4 plants. And that can be really useful for situations where we're trying to tease apart how much C3 versus how much C4 organic matter organisms might be using. And I'll give an example of where that's relevant moving on. But one big piece of this that you may not already know is a lot of the plants we care about out in the natural world are going to be C3 plants, and a lot of the foods that we eat as people are going to be processed with things like corn syrup or cane sugar, so there'll be a large C4 signal there. So in many studies where folks are interested about whether anthropogenic resources are being used by animals, this C3, C4 difference can be quite powerful. But I'll expand on this example a little bit to say that the plant world isn't so simple as just, you know, Rubisco and PEPcase out there doing things. I've now rotated the axis such that the very low values on the delta 13C scale are near the bottom of your screen. More positive values or less choosy plants are near the top of your screen. You can see you've got your C4 plants and your C3 plants here. There's a group of plants we call the CAM plants, the crassulacean acid metabolizers, which kind of have aspects of both plants. So you see them span the whole gamut of carbon isotope ratios. And we also see a range of carbon isotope compositions being shown in aquatic vegetation. So seagrasses are organisms which use actually not atmospheric CO2 a lot of times. They might be pumping in bicarbonate from ocean water, which has a very positive isotope composition relative to atmospheric CO2. So you might see seagrasses being much more positive than many other plants we might measure. Kelp might be using dissolved CO2, but also pumping some bicarbonate. They may be CO2 limited, so they aren't able to be as choosy in the long run. In general, freshwater plants, most of the freshwater plants that we interact with as freshwater ecologists and things of that nature are using a C3 type strategy. And they have a slightly wider range than C3 plants because a little bit more factors into what actual fractionation or how choosy they're being at any given time. And some of those factors are shown here on the right on the terrestrial vegetation side of things. So increases in light, increases in temperature, increases in the amount of nutrients, they generally make plants what I would refer to as less choosy. They reduce the fractionation that you see. And the way that you could imagine that is if, again, using this analogy of Rubisco being, a person playing with Legos, if you're in a real hurry to build something, you may not have the opportunity to dig through your entire bin of Legos looking for the exact color of Lego you want. You may just have to grab the first Lego that works that comes to hand and keep on fixing carbon, as the case may be. So while photosynthetic strategy is really important and physiology is really important to shaping what we see in the carbon stable isotope composition of things, environmental conditions are also playing a big role in this stuff. And that can be an important piece of this when we think about differentiating aquatic zones. So many people might be interested in studying a population of fish, and they're curious about whether or not this fish is feeding primarily in littoral zones. Are they hanging out in the pelagic zone? Are they feeding on benthic organic matter primarily? And we know from previous work by France and others, or I think this was a single author publication, so I should say by Robert France in 1995, he analyzed a number, well, approximately, I guess, do the quick math, approximately 400 samples, 412 samples of aquatic producers from 4 Canadian shield lakes, and found that by differentiating, he had separated them by pelagic and littoral beforehand, and he found that the pelagic and littoral populations of producers separated themselves quite nicely, a mean of about -24 for the littoral producers, a mean of about -34 for the pelagic producers, which is a really good foundation to separate consumers that you might think are using primarily pelagic or littoral organic matter. One issue, if you were trying to do work like that, as you can see right around that -32 to -28 zone, there is significant overlap of the carbon stable isotope compositions. And this is a recurring problem in stable isotopes. A lot of times folks question about stable isotopes is, can I get down to the taxa level? How well can it really differentiate things? And the answer is sort of a non-answer because the reality is isotopes can differentiate practically anything. They can differentiate anything, so long as what you're trying to differentiate is isotopically different, which in many cases, they will be isotopically different from each other, but in many other cases, you may have overlap that makes it challenging to make the differentiations that you'd like to be making. I've already kind of touched on this a couple of times, but I just want to drive home the fact that, again, why stable isotopes are useful is we find that stable isotopes vary for a wide variety of reasons related to a wide variety of environmental factors, benthic pelagic gradients, inshore, offshore, aquatic, terrestrial, you name it. There's probably a system with which you'll find usable isotopic variability. And if not carbon isotopes, maybe you're looking at other isotopic systems, which I'll discuss a little bit later. But in general, stable isotopes vary based on so many factors that there's usually some useful information to be extracted from isotopic information, as long as you have a strong enough conceptual model to tease apart exactly what you're learning. So I've talked primarily about photosynthesis and things of that nature, which is important for the kinds of consumers that most of us that care about things like salmon are interested in. But really, again, we're interested in consumers. We want to know what's going on with the salmon. So how does that information about what Legos plants are using factor into what information we can get about what Legos consumers might be using or what foods consumers might be eating? And the kind of quick and dirty to this is you are what you eat isotopically. This is a statement that was coined quite a long time ago. The general idea being if you go and eat a plant that is made up primarily of black Legos, you will build tissues using the organic matter from that plant that is black Legos, vice versa with white and blue Legos. So you are what you eat. If you are eating resources that have a set combination of isotopes in them, you will take on a similar isotope composition as the resources you're using. It's not good enough in science, as many of you are probably familiar with, to say, oh, it's black Legos, it's blue Legos. A lot of times, we're forced to put some numbers on these things. A common thing you'll see in stable isotope studies is the use of stable isotope mixing models. And I'll give, again, this is a pretty generalized, oversimplified version of this. We've now got some pretty cool, complex computational tools to really drill into these data. But in general, stable isotope mixing models are like imagining mixing of paint. If you have just 1 times yellow paint, 1 times red paint, you might have a sample at the end that's orange. And If you mix twice the amount of yellow, you might have a slightly more yellowish orange. If you have twice the amount of red, you might have a slightly reddish orange. And so isotopes work the same way. If we're eating a mixture of food sources that have two separate kinds of isotopes, we'll be an additive sum of those two pools of isotopes, and we can back calculate. If we know what the composition or what the color of the original sources were, we can back calculate how many of each of those sources are in our mixed pool of them. One thing to be careful of in these models, though, is over-parameterized models. So a lot of times people will go out and they'll sample every potential food source that an organism might be consuming. So what you'll end up with is 5 shades of yellow and 5 shades of red. In that situation, it becomes really difficult to tell which of your yellows and which of your reds was contributing. So that's a situation where it might be best to start pooling some of your sources, or really considering whether or not all of those sources that you collected originally need to be included. So improve your conceptual model to rule out some of the potential sources. It can also be a problem if you're missing a source. One of the evils of stable isotope mixing models is they will always give you an answer. So if I had, say I thought my sources were a mixture of yellow and red paint, and I go analyze my consumer and I get a purple consumer, it may tell me that that's a 50-50 mix of yellow and red, just because models being models have to spit out an answer. But in fact, it's pretty clear that the bucket of purple paint that I have, this consumer stable isotope composition, couldn't have possibly come, or couldn't have reasonably come, from a mixture of the yellow and red sources that I have, the two sources that I've analyzed beforehand. So it's really important that you haven't missed any sources. And there can be signs, obviously analyzing a consumer that has a very different composition than any of my sources would be a red flag, but it can't always be so clear. So you really need to be sure that you have every important source. And lastly, this sort of falls under the same argument as the over-parameterized models, but be aware of overlapping sources. You can't differentiate things that aren't isotopically differentiated themselves. So I'm getting a little behind time, so I'm going to rush through this real quick. This is just one of the original examples of demonstrating that stable isotopes mix conservatively. This is some work by Brian Fry in 1978, where he did the painstaking work, I cannot imagine doing this, of extracting gut contents from grasshoppers, identifying the gut contents of those grasshoppers down to, I think, as near to species and taxa as he could, definitely differentiated between the abundance of C3 and C4 plants in the diet. He analyzed all those C3 and C4 plants on the landscape. And here on the y-axis of this plot, you see the carbon stable isotope composition he analyzed in the grasshopper itself. And here on the x-axis, you see the carbon stable isotope composition that he calculated based on what he assumed to be the mixture of plants in their guts. And what you can see is this is a line with an intercept near 0 of a slope almost near 1. So by comparing the prevalence of C3 and C4 plants in the guts, to the values that he analyzed in the grasshopper tissues, he found that the contribution of C3 and C4 plants to the diet was exactly shaping the composition of carbon isotopes in that organism's tissues. So really quickly, I'll gloss over this. I'm going to move on to nitrogen stable isotopes in a minute. Really, carbon stable isotopes are great for characterizing different sources of fixed carbon if you're interested in whether or not algae are contributing versus terrestrial plants, C3 versus C4. It's carbon isotopes. Obviously, it's going to be good at looking at things that have to do with carbon. You can also identify where inorganic carbon sources are being used. Oftentimes, inorganic carbon can give you a funny sort of carbon stable isotope composition. I'll get into an example where it's used to study trophic structure in a minute. And also in systems where methanotrophy is important. Reduction of carbon dioxide to methane that might be released to the atmosphere or be reconsumed by organisms. You also get a very funny carbon stable isotope composition. It's a bummer, or a downside of carbon stable isotopes, that many of the sources overlap. So you won't always be able to tease apart the things you want to, and that not all variability is going to be meaningful to you. And I'm going to gloss over that last bullet point here in lieu of diving into some of the information about nitrogen stable isotopes. So carbon stable isotopes, ecologists and environmental scientists, a lot of what we think about through time is where is production happening, what sort of producers are contributing, how is photosynthesis occurring. That's what really drives a lot of the useful carbon stable isotope variability in the environment. A lot of that stuff factors into, microbes and plants doing things to nutrients, but definitely in many environments, there's a much greater control on the nitrogen stable isotope composition that you'll measure coming from where exactly the original source of nitrogen in the system is. In heavily fertilized systems, the fertilizer tends to have a value near 0. you'll find that those systems tend to have values near zero in their producers. In systems that have been fertilized with animal manure or wastewater, I shouldn't maybe say fertilized with wastewater, but in systems that are getting their nutrients from those kinds of places, you'll find they have much more positive nitrogen stable isotope compositions. And the inherent starting point of any given ecosystem in nitrogen stable isotope space, it may have implications, based on the source of those nutrients or something, ecologically speaking, but it isn't inherently meaningful. That baseline point that you start from, the original nutrient that makes its way into the food web, it could be 0, it could be 10. That may be diagnostic for telling you whether or not the primary nutrient is mineral fertilizer or animal manure, but it really doesn't matter to anything higher up on the food chain. So I'll address this idea of a baseline a little bit further as we move forward, but that's just one point I wanted to make is the sort of starting point in any given ecosystem has a lot to do with where those nutrients are coming from. And once you've identified that starting point, the actual exact point you find yourself on at the delta 15N scale, a lot of times is not all that meaningful. It's the differences that you start interpreting at that point. So I'll dive into exactly what sort of processes will alter nitrogen isotopes in a system. This is an example from some work done by Carol Kendall and others. There's another slide further down that shows some more of our work looking at transformation of ammonium, which is one of the primary nutrients you might find in a river system, traveling downstream through the Sacramento River. And we might have an original pool of ammonia that is, I'm going to use this Lego analogy again, a mixture of red and blue Legos. And this red arrow there is a process called nitrification. And it's a microbial process where the microbes will take ammonium and oxidize it to nitrate, and there will be a residual pool of ammonium because rarely in nature, right? I shouldn't say rarely in nature do we see everything react, but a single microbe would find it hard to react the entire pool of ammonium. And when that nitrification process occurs, the microbes tend to prefer the lighter isotope of nitrogen, that is. So the more red Lego containing nitrate or ammonium will react a little bit more quickly than the blue Lego containing ammonium. And so what you'll find is that the initially produced nitrate will be slightly lighter in isotopic space, or will have less of the heavy isotope than the original pool of ammonium, and the ammonium that's left behind will be slightly heavier. Because if you have a pool of Legos and you're slowly only pulling out all the blue ones, eventually you'll just be left with the red ones, or vice versa. And this process can continue on and on with the residual ammonium becoming more and more enriched, and by enriched I mean to say having more positive delta 15N values as time goes on, as you remove more and more. I flopped it in my last example, but I guess that in this slide, you're removing the red Legos in the process of nitrification. Uptake is similar. Plants will tend to uptake the lighter isotope slightly more quickly than the heavy isotope. So if you had two pools of algae that were growing on nitrate and ammonium exclusively, you would find that the algae that grow on the ammonium exclusively would have this value very much like ammonium, although slightly lower in delta 15N space, while those growing on the nitrate would have a value like the nitrate, but slightly lower in delta 15N space. So they took this a bit further in the work by Kendall and others, and they did a longitudinal study of the Sacramento River. And this is an odd slide because you do have to read it right to left. That x-axis there is river mile moving from right to left. That would be inland to the negative values are going out into the estuary just about. And the very bottom where you see the filled area here is the concentration of nutrients. So as you move downstream, this large spike in the ammonium concentration is the Sacramento River wastewater treatment plant. They're releasing ammonium into the river. And we see a relatively stable amount of nitrate in the river, although it grows as we see the concentration of ammonium going down. And that would, of course, be the impact of nitrification. We're removing ammonium from the water column and adding in some nitrate. So I'm going to move higher up onto the axes here. This is a pretty complicated graph. And now these lines, the lines are the nitrogen stable isotope composition of those pools of ammonium and nitrate. And really the one that does the most interesting thing in this plot is the ammonium. What we see is it's pretty flat during the period where there's tons of ammonium in the water column. But as you start to remove ammonium from the water column through the process of nitrification, you're removing the lighter stable isotope. So you're seeing more positive values in the residual ammonium, more positive delta 15N values, more of the heavy isotope left behind because you're removing the lighter stable isotope during nitrification. And you may think to yourself, oh, biogeochemistry is cool, nutrient cycling is cool, but again, I'm interested in salmon. What does this have to do with me? And most of you are probably familiar with the idea of salmon being anadromous fishes in many cases. So if you had an organism that was migrating or moving through this stretch of river, the difference between analyzing them and thinking they're in a food web that has a delta 15N value of 14 and analyzing them and knowing they're in a food web that has a delta 15N value of about 6 would be a misestimation of the trophic position of those salmon. An estimation of trophic position is one of the key or most common uses of stable isotopes of nitrogen. That'd be a misestimation of the trophic position by about 3 trophic positions. So without understanding how the biogeochemistry and how nutrient cycling could be affecting the nitrogen stable isotope numbers that you're measuring, you could make some really grievous ecological missteps in interpretation. So I just mentioned that nitrogen isotopes are used to look at trophic position in many organisms, and that's because in delta 15N we see a generally a pretty large increase with each increasing trophic level, and that's because whenever we eat anything, we say eat a sandwich that has some carbon and nitrogen in it. I'm going to use some of that carbon for energy, and I'm going to use some of that carbon for energy. I'll start there. I'll use some of that carbon for energy, and I'm going to exhale some CO2, as most of you are familiar with. Our breath contains CO2 in it from the oxidized organic matter that we've eaten. The process of respiration is going to preferentially lead to excretion of 13C. I'll leave it at that. And so what's left behind in pardon me, actually this slide is backwards. So I wrote the wrong thing on this slide. We're preferentially excreting the 12C and the 14N. So we preferentially excrete, breathe out, or it goes out in our urine after going through the urea cycle if it's nitrogen, the lighter isotopes of carbon and nitrogen. So what gets left behind in our tissues is slightly heavier isotopes. So if I ate only sandwiches that had a carbon stable isotope ratio of about 0 and a nitrogen stable isotope of about 0, my carbon stable isotope ratio and my nitrogen stable isotope ratio at trophic position 1 above my sandwich here would be about 1 per mil for carbon, and for nitrogen about 3 per mil. So we're about 3 per mil in nitrogen per mil space than our food sources, and about 1 per mil in carbon isotope space than our food sources. We don't have a ton of data about sulfur right now. It seems to be a variable fractionation or trophic diet or diet tissue offset that we observe in sulfur, but it tends to be near 0 is what we're thinking. So I'm going to jump past this again. I'm falling kind of significantly behind schedule at this point to get into a case study where this idea of a baseline becomes important. And this is some work by McClellan and others where they were looking at the ecology of a estuary embayment near Nantucket Sound. And they were curious about using nitrogen stable isotopes to put together a food web, a trophic ladder for this system. So they went out, and an obvious place to start is to analyze all the producers in their system. And they found just within this small system, a range of about -2 to +6 in the primary producers. We've got Zoster marina, which is an eel grass, some plankton. You've got your cord grasses here. This is a salt marsh grass, pardon me. And I think Vagabunda is a bug, but these other ones are planktons and algae and things like that. So while they all might have access to a similar sort of subset of nutrients, we still might see this variability, 4 to 6 per mil, 0 to 4 per mil, just based on the physiology of uptake, the biochemistry that takes place inside the organism, and maybe they are using slightly different amounts of different nutrient sources. So even in co-located plants, you can see some variability in their nitrogen stable isotope composition. The next step, since they wanted to build up the food web, was to go analyze some consumers. We got the Heterocletus here, which is the mummichug, which is a pretty common fish on the East Coast there. But they found a pretty broad range of nitrogen stable isotope compositions in their consumer species there at the top. This range of about 6 to maybe 12 per mil, 8 to 12 per mil, that suggests that these fish, and Obscura is a bug, pardon me, it's a winged insect, maybe spanning 1 to 2 trophic positions, I guess closer to 1 trophic position in the system, which is a pretty broad range of ecological things to be doing if you think about eating at 1 trophic position and then occasionally eating at 1 whole trophic position higher. And so you could have an ecological interpretation to that, which is, yeah, these fish span a whole trophic position. But there's also another possibility. I mentioned at the very beginning of this nitrogen stable isotope segment that the actual starting point in nitrogen stable isotope space is sometimes meaningful, but for applications like this is not always entirely meaningful. So you could have two basically identical food chains that just kind of start a different place. And we would call this a baseline difference. So the nutrients that are making into primary producers have a different nitrogen stable isotope composition. Nothing in a trophic position above the producers would know about that. And they just keep building with their trophic discrimination, moving up into more positive nitrogen isotope values after that. This top predator with a value of 15 isn't actually at a higher trophic position than the one that had a value of 13. It's just that the nutrients at the base of the food web had a different value to start from. And that's indeed what they found when they increased the sampling of their, and I say increased, I'm presenting this a little bit narratively. When you read the paper, it's all just presented to you like this. So I'm taking a little bit of artistic liberty here. But increasing the sampling, you can see that in the more urbanized part, where wastewater is likely contributing to the nutrient pool, you find these more positive values in all of the same producers. But out in this Sage Lot Pond site, which is a more naturalized site, you have lower values. It's the same organisms doing the same things. They just have access to nutrients that have a slightly different starting point. So you build that food web up in those two habitats from a different starting point, but the food chain may in fact look basically identical. So we can see here that the original set of data that I showed you, we see primary consumers and secondary consumers going up nice and linearly, increasing in their nitrogen stable isotope composition with increasing trophic level. Now that I've added in that second set of samples that came from the more urbanized site, we can see that there's indeed a second set of consumers that follow along that more urbanized nutrient pathway. So going to wrap up really quickly to say nitrogen isotopes, they're great for characterizing trophic structure, determining nutrient sources, studying nutrient cycling if you're interested in things like that, but you need to be really wary of biogeochemical artifacts and new nutrient sources. So biogeochemical artifacts being transformation of nutrients that alter stable isotope ratios in ways that you might not have expected, and new nutrient sources being a source of nutrients that has a funny isotope composition that you hadn't counted on affecting your data. I presented the carbon and the nitrogen fairly separately up to this point, but really in many cases, you'll see them combined. A dual isotope approach is really powerful because it might allow you to, at the same time, separate out a food web that's based on planktonic organic matter here in the more negative values and benthic organic matter in the more positive values, and then look at increasing trophic level above those. And you may have a channelized system where you're going directly up using only one source of primary production, and then one of the big ecological roles of top consumers is integrating multiple pathways. So you may have a top consumer up here that has an intermediate carbon stable isotope ratio, pulling in some production from two different productive pathways, but is serving as a top consumer at a very high trophic level in the system. You can also use it to see when fish that are living in a certain habitat aren't using the resources you think they might be using. So this was a study that was done by Duckmark and others in Chile, looking at benthivorous fish. So fish that are living on the bottom, feeding on the bottom, you might expect that they're eating organic matter that comes from the bottom. But in fact, carbon isotopes and nitrogen isotopes indicate that these fish are primarily consuming organic matter that's sinking down from the pelagic zone. So while these fish live in a benthic environment, if they were feeding on benthic resources, you might expect them to all plot here. They are in fact feeding on resources that are coming down from the pelagic environment. So stable isotopes can be a really powerful tool to detect when organisms are doing funky stuff like that, because it doesn't matter where they spend their time. It really matters where they're getting the organic matter. Where are they getting their Legos? Here's an example from Lake Michigan by Eglite and others, pardon me, where they were able to show that fish living in river mouths on Lake Michigan were getting subsidies from both the lake and the river, whether or not they were living in the lake, river, or vice versa. So on the x-axis, you have whether the fish was taken in the lake or river site. The colored boxes are all organic matter that would be coming from, or it's the estimated contribution of organic matter from the lake. And the white boxes are all the estimated organic matter that's coming from the river. They used actually a 3 isotope model here, carbon, nitrogen, and I believe it was hydrogen. So, again, using some of those more complex computational tools that I mentioned. So a 3 isotope model perhaps. But the important thing here is in almost no sites do you see the lake having 100% resource use from the lake and the river having 100% resource use from the river, we see that the fish are getting their energy from varying sources, a dynamic source, whether or not they're in the lake and the river. So isotopes are really powerful for, again, showing you when organisms might be getting energy from places that they're not exactly spending a lot of time. Another really great application for this sort of work is a lot of times we assume, pardon me, what am I trying to say with that? In the example I gave before, we were trying, that is the Wakiat Bay example, we were trying to understand trophic structure. And the fact that carbon and nitrogen isotopes varied spatially was noise in that relationship. It made it hard to see what we wanted to see. We can flip that relationship on its head and say, let's assume that our organisms are doing ecologically similar things wherever they are, and let's see if the stable isotopes vary across space. So this is some work by Espinal and others in 2002, where they built what was called an isoscape, or an isotopic map, and using carbon and nitrogen isotopes for the North Atlantic. And they found that there were these large kind of zones that they could delineate that had relatively consistent carbon and nitrogen stable isotope compositions through time. And a lot of clever modeling and consideration of things like sea surface temperature went into play in this. So I don't want to oversimplify too much, but I do want to just present the example of now, if we assume that, say, a salmon that might have free range across the North Atlantic is going to be at a similar trophic position wherever they are, we could compare the values of isotopes that we measure in those salmon and potentially estimate where they were spending their time out in the North Atlantic. So my bit at the top there about one model's confounding variable is another signal is about flipping that relationship, where sometimes spatial variability in isotopes make it hard to see ecological structure that we want to see using isotopes, but sometimes if we make some assumptions about ecological structure, we can use spatial variability in stable isotopes to learn something different about our study organism. And so I am running over time. I'm going to just absolutely blast through this last bit to point out that there are, in general, isotopically, two kinds of tissues. We think of it about inert tissues and metabolically active tissues. Inert tissues are what I'll discuss really quickly. Inert means that they're conserving their composition during the time of synthesis. So inert tissues are things that aren't metabolically active. Once we've built that tissue, once we've put isotopes into it, we're not taking them out. Things like hair, things like tree rings, things like salmon otoliths are inert tissues. So if we go out, find feathers in the world, extract salmon otoliths and archive them, extract salmon scales and archive them, core trees, we can learn things about what was happening when that tissue was originally synthesized way back in time. This has been done with salmon eye lens layers. So for those of you who don't know, fish eyes grow like onions, so you can peel off layers. So on the far left of this plot, we have the early stage when the fish are living in the hatchery. We have the estuarine stage in the middle lamina, and you can see them moving to use of marine resources, which have much more positive values in the outer eye lens layers. A quick touch on sulfur stable isotopes. This is analysis of otoliths from salmon, where they were looking, you have to read this plot from right to left too. Hatchery fish are eating marine chow that has a very positive sulfur stable isotope composition. If you have a fish that leaves the hatchery and stays in the river, it stays at a fairly positive sulfur stable isotope composition throughout the concentric rings. They grow just like tree rings. But as a fish that gets older, immigrates away from the hatchery, I should say, down the river, if they go onto a floodplain habitat, floodplains have some funny biogeochemistry happening on them that gives the sulfur organic matter there very low delta 34S values. So we can see use of those floodplain habitats in the otolith data going way back in time by archiving otoliths and taking measurements moving across that otolith. And there's metabolically active tissues. This plot on the right, that shows the approximately every single day our cells are dying and being replaced. On the whole, about 65% of them are this erythrocyte type cell. We're losing about 1.1% of our skin cells every day. So all this to say, staying alive involves cells dying, we get rid of their organic matter, and we replace it with the new stuff. We can see this if we do what's called a diet switch study. This was done with yellowtail by Madigan and others, where they had fish that were fed a pellet diet, then they switched them to a fish diet. And you can see they moved from having a composition, a nitrogen stable isotope composition with a little bit of offset, very indicative of eating a pellet diet, so a very low value, they transit towards a much more positive nitrogen and carbon stable isotope value. And you can see that turnover happens at different rates for carbon and nitrogen isotopes. So again, that might be a problem for you if you're trying to detect only a transitory signal, but that noise in the signal can also be the signal you're looking for, because these signals might be incorporated into different isotopes, into different tissues at different rates. So something like liver, you can see it's a very steep line. After the diet switch, the liver, within 20 days, it looks like, has come to the new value, whereas a tissue like, say, red blood cells or muscle, even out at 100 days, there's still significant slope in the line. The value's still changing. So not all of the old muscle has been replaced by the new dietary source yet. And one last thing, we can interpret that to mean if certain tissues have come into equilibrium while others cannot, that can help us nail down how long it's been since that dietary switch. So we can build what's called an isotopic clock. This was some work by Shipley and others who are looking at the immigration of sand tiger sharks into estuaries. And they were able to use one of these isotopic clock models to estimate the time since estuarine arrival of individual sand tiger sharks. And so finishing up here, again, sorry for going so out of time. What else is out there? Things I didn't get an opportunity to touch on, which are totally exciting topics in and of their own. Many of you may have heard of compound-specific stable isotope analysis. That would be looking at specific molecules in tissues like fatty acids or amino acids. This is a really powerful tool for food web reconstruction and trophic level estimation, because actually you're able to internally correct for those baseline effects when you're making compound-specific measurements. There's hydrogen and oxygen isotope systems, which I didn't touch on at all here, but they're equally interesting in their own right and let you study a number of cool things. You can make gas measurements of isotopes. Those can teach you cool things about greenhouse gas emissions, the metabolic rate of organisms, things like that. And everything I've talked about here has been what would be colloquially referred to as light isotope systems. There's also a whole world of heavy isotopes. There's folks that are looking at zinc isotopes, trying to learn about trophic level stuff, looking way back in time, ancient shark teeth. You can use strontium isotopes, which is a heavy mineral that can teach you about what parts of a river drainage salmon are spending their time in. And so these heavy isotope systems, there's a lot of promise in those too. If you think isotopes are really interesting and you want to learn more, there's a couple of resources here. I would particularly recommend this book, Stable Isotope Ecology by Brian Fry, who I showed that example from earlier. It's an extremely approachable text. It's extremely fun to read in many ways, which is not something that can be said of many other textbooks. And it'll give you a really great foundation to kind of understand what isotope people are doing out there in the world. And some of these other texts are a bit more technical, but can really get your feet on the ground with respect to how isotopes are used in the world and what the real mechanics of isotopes are, get into the nitty gritty. So with that, I'd like to thank everybody for your attention. Sorry for kind of going over time and having to rush a little bit here at the end, but I hope you've all been able to take something valuable for you away from this. And I'm happy to take any questions.
Awesome. Thank you so much. That was an excellent presentation, Bobby. Yeah, so we will now open the floor to questions. So if you have a question, please raise the little hand on your screen and I'll be able to unmute you and introduce you to ask your question. This will be the preference, but if this function does not work for you, you can type your question in the chat and I'll read it out loud. See if any questions come in. All right, so if there are no questions, give it another second. Okay, well, thanks, Bobby. So if you do have a question later on, please feel free to reach out to the foundation and we'll pass the question along to Bobby. We have recorded this session and it will be available on our YouTube page if you would like to see that after the fact. And I also want to remind everyone that our next webinar will be in the new year on January 22nd with Robert Lennox, Associate Professor and Ocean Tracking Network Scientific Director from Dalhousie University. The title of his webinar will be Behavior, Survival, and Marine Ecology of Atlantic Salmon Kelts. Where are the repeat spawners? So again, a big thank you to Bobby and to everyone who participated. We will hopefully see you in the new year. Bye now.
Bye now. Thank you.